Everyone. First off, I'd like to thank the ISMB SysMod organizers for inviting me to this talk. And today I will be presenting an agent-based model called CellAgentChat for deciphering cell-cell interactions from single-cell data. So in multicellular organisms, cellular activities rely on the coordination of cell-cell interactions across various cell types and tissues, facilitated by ligands and receptors. Cell-cell interactions are critical in numerous biological processes such as cell differentiation, disease pathogenesis, and immune response. Inferring cell-cell interactions from in biological processes is challenging due to cellular heterogeneity within tissues, where each cell type has unique interactions and signaling pathways. Cells release diffusible ligands into the environment that can bind to receptors and trigger various responses in cells. For instance, a ligand might bind to a receptor and initiate a downstream signaling response. I don't know if you can see my pointer. No?
You can do right click.
Okay. Yeah. Sorry about that. Okay. So cells can release diffusible ligands that can trigger nearby responses. So for instance, a ligand might bind to a receptor and trigger a downstream signaling response, or the receiver cell may not respond at all. Spatial information is also important for cell-cell interactions. So cells in closer proximity are more likely to interact, whereas cells that are more distant make ligand-receptor interaction more difficult. Despite these challenges, advancements in single-cell RNA sequencing and spatial transcriptomics offer unparalleled opportunities to investigate cell-cell interaction at high resolution. Single-cell RNA sequencing allows us to analyze the gene expression profiles of individual cells across all genes. Spatial transcriptomics, on the other hand, provides spatial context to gene expression data, mapping where specific transcripts are located within a tissue section. Together, these two techniques enable a detailed understanding of cell-cell interactions. Numerous tools have been developed in recent years for cell-cell interaction inference. Despite advancements, earlier methods and versions primarily rely on single-cell RNA sequencing data, lacking spatial distance information, which limits their ability to infer cell-cell interactions accurately. Since cell-cell interactions in tissues are heavily influenced by spatial structures and occur at various ranges, integrating spatial information is essential for studying long-range and short-range interactions. Additionally, some methods only identify interactions at the cell population level. This means they group cells into different populations and infer interactions between these groups, thus ignoring individual cell heterogeneity. Finally, despite some methods' ability to capture the effects of cell-cell interactions on downstream genes, they don't support in silico perturbation. In silico perturbation allows for the simulation of experimental conditions to explore potential therapeutic targets. Our approach for inferring cell-cell interactions uses agent-based modeling. Agent-based models simulate the actions and interactions of autonomous agents. Agents are typically individuals or entities endowed with specific characteristics, behaviors, and rules. In the context of cell-cell interactions, each agent is a cell. These agents can then interact with each other and the environment around them, leading to emergent behaviors at the macroscopic level. Now, although agent-based models hold great potential, to our knowledge, they haven't been fully utilized for tasks involving single-cell data. So our model, CellAgentChat, is an agent-based model. Utilizing the principles of agent-based models, we characterize each cell agent through various cellular attributes, which include the gene expression profile of the cell, the spatial coordinates of the cell within a tissue from spatial transcriptomics data, the cell type information, and the ligand receptor universe from various databases. So the initial step in our approach involves the estimation of the receptor receiving rate alpha. This is the probability that a ligand is received by the receptor of the receiving cell. So here we employ min-max normalization to calculate the receptor receiving rate for each receptor in each cell, thereby establishing both a receptor and cell-specific metric. Then, in order to account for the effect that cellular interaction have on the downstream gene expression, we employed a neural network to estimate the impact of cell-cell interactions involving each receptor on the downstream gene expression dynamics versus when we block that receptor in silico. This is known as the receptor conversion rate beta. The neural network used comprises of two input layers, one for ligands and one for receptors. We also have two hidden layers. The first one represents the ligand receptor pairs, where we have a connection from one receptor input and one ligand input. And the second hidden layer represents transcription factors. Finally, the output layer aims to predict the expression of all genes in the cell. The connections in the neural network between the ligand receptor pair layer and the transcription factor layer, as well as the transcription factor layer to the downstream gene layer, are informed by receptor transcription factor interactions and transcription factor gene interactions from various databases. So we use prior knowledge to get these connections. We quantify cellular interactions between a sender and receiver cell based on the number of ligands secreted by the sender cell and subsequently received by the receiver cell. This process hinges on three interrelated components: the receptor receiving rate, the receptor conversion rate, and the ligand diffusion rate. The ligand diffusion rate estimates the rate of diffusion of a ligand. This rate is a function of both ligand expression and intercellular distance. It is also governed by two parameters, tau and delta. Tau represents the degrees of spatial freedom concerning spatial transcriptomics data and is typically set to 2 for spatial transcriptomic data that is derived from two-dimensional slices. Delta signifies the decay rate of ligand diffusion, which prioritizes interactions over long or short ranges. When delta is lower than 1, it weakens the decay rate, giving precedence to longer range interactions. Conversely, when delta exceeds 1, it amplifies the decay rate, giving preference to shorter range interactions. When delta is set to 1, which is the default setting, it equally prioritizes long and short range interactions. We then quantify interactions between a specific ligand receptor pair, between a sender and receiver cell agent, using the three rates that I mentioned. And then we can also get the score for two specific cluster pairs, a sender and receiver cluster pairs across all the cells for a single ligand receptor pair. And then we also get the score between two cluster pairs across all ligand receptor pairs as well. CellAgentChat also calculates cell receiving score, which is which measures the received interactions for each individual cell. So to evaluate the performance of CellAgentChat, we undertook a comparative analysis of CellAgentChat against 5 prominent cell-cell interaction inference methods. So first, we assessed the cell-cell communication networks generated by CellAgentChat and compared them with the outcomes of other methods. However, due to the absence of a definitive ground truth, we decided to adopt a consensus approach. This involved normalizing the interaction matrices computed by each method and averaging them together to create an ensemble interaction matrix serving as our new consensus reference. We then computed the Pearson correlation between the newly calculated ensemble interaction matrix and the calculated communication patterns from each method. We found that CellAgentChat was comparable in performance with state-of-the-art methods CellChat, niches, and Komoot as well, but displayed much lower variability than these other methods and therefore was more robust across all data sets. To validate that the cell-cell interactions inferred by CellAgentChat are spatially informed, we computed the Pearson correlation between the predicted communication matrix and the cell-cell inverse distance matrix. We grounded our analysis on the established understanding that cells that are closer in proximity are more likely to interact. Cells predicted by CellAgentChat as highly interacting were close in proximity to a similar level as niches and Komoot. And again, we showed low variability here. Finally, we proceeded to conduct pathway enrichment analysis to further confirm the biological relevance of the inferred ligand receptor pairs. We compared the enrichment p-values for relevant pathways using ligands and receptors from the top 100 significant ligand receptor pairs from each method. The pathway enrichment analysis for CellAgentChat was in general better than other methods as seen in the statistical significance scores at the top of the plots here. Now, although our evaluation shows that CellAgentChat predicts cell-cell interactions at a similar level to state-of-the-art methods, it is our various downstream functionalities that make CellAgentChat stand out as a useful tool for researchers. One of our key functionalities is in silico perturbation to identify receptor antagonists that could serve as suitable therapeutic targets for cancer and other related diseases. To showcase this functionality, we use CellAgentChat to build upon previous research in breast cancer, aiming to identify significant receptors related to the disease and find therapeutic approaches. So first, we use CellAgentChat to pinpoint key receptors involved in the top ligand receptor pairs, and we identified 13 key receptors here. We then simulated the blocking of these receptors individually, akin to the action of receptor inhibitor drugs. So we deployed the neural network that was previously designed to calculate each receptor's conversion rate to predict the impact that blocking a receptor would have on the downstream gene expression, and identified the most affected downstream genes for each in silico blocked receptor. Our analysis with CellAgentChat indicated that blocking 9 of the 13 receptors, as identified in red here, significantly disrupted genes associated with breast cancer. Now, although all 9 receptors identified are linked with breast cancer through various other studies, some of the identified receptors were particularly more interesting to us, such as EGFR. So CellAgentChat identified that blocking EGFR significantly disrupted genes associated with breast cancer. So in particular, when EGFR was blocked, TPD52 over here was the 5th most affected gene and was previously linked with breast cancer. Similarly, another gene, HMGA1, which is also associated with breast cancer, was the 6th most affected gene. These genes notably influence immune cells such as mast and macrophages, aligning with the context of breast cancer where immune cells play a crucial role in the response against cancer cells. We also conducted a survival analysis of breast cancer patients over time, which revealed statistically significant differences in survival probabilities for patients with varying levels of expression of EGFR. These findings indicate that the receptors identified by CellAgentChat would serve as suitable therapeutic targets for treating breast cancer. In fact, there already exists an FDA-approved drug designed to specifically target and disrupt the pathways involving EGFR in breast cancer, which act as support to our findings. We also have similar results like this for the other 8 receptors we identified as well. Now, CellAgentChat can also be used to identify long and short-range interactions. This is one of our other downstream functionalities. So our understanding of the specific distances at which ligand receptor pairs operate is evolving but remains incomplete. The agent-based model employed by CellAgentChat facilitates identifying both long-range and short-range interactions by adjusting the delta factor associated with the distance between cells. So to identify short-range interactions, we used a delta of 10 to constrain interactions to shorter distances, and we used a delta of 0.1 to identify long-range interactions. To validate our results, we used the CellChat database, which provides known annotations of the distance ranges of ligand receptor pairs. So ligand receptor pairs are either categorized as secreted signaling or cell-cell contact. So secreted signaling is more long range, cell-cell contact is more shorter range. So under our short range mode, where delta is set to 10, we identified that 80% of the top 15 ligand receptor pairs were actually annotated as cell-cell contact. So in green here is cell-cell contact, red is secreted signaling. So 80% of the top 15 was cell-cell contact. Meanwhile, in our long-range mode, where delta is 0.1, 75% of the top 15 ligand receptor pairs were classified as secreted signaling. Now, compared to another approach that is based on optimal transport, CellAgentChat more accurately captured the short-range interactions and identified long-range interactions with a similar degree of accuracy. The ligand receptor genes associated with short-range interactions identified by CellAgentChat showed enrichment in pathways related to cell adhesion and cell-cell junctions, which are known to occur at short distances. In contrast, the ligand receptor pairs associated with long-range interactions were enriched in numerous signaling pathways known for their wider regulatory range. Therefore, CellAgentChat can be used to accurately identify long and short-range interactions. So to conclude, we developed CellAgentChat, an agent-based model capable of modeling cell-cell interactions from single-cell data with various downstream functionalities, such as in silico perturbation and identifying long and short-range interactions. This study establishes an initial framework for employing agent-based models to model other cellular behaviors using single-cell data. So on the slide, you can see a QR code that goes to our preprint of our paper on bioRxiv, and then also the GitHub link is there as well for anyone to try out our tool. And I'd just like to acknowledge my supervisors, Dr. Jun Ding and Dr. Yue Li at McGill University for their continued support on this project, as well as our various collaborators and funders. Thank you so much.
Thank you, Vishwak. That was great. Any questions in the room for Vishwak?
Great talk. You talked about the spatial technologies, and most of them actually are not at the single cell resolutions. So how do you think this model, how does that impact your results in general?
Yeah, that's a great question. So as you mentioned, there's lots of different technologies. Some are at the single cell resolution, such as like SeqFish, for example. So we have like data, we used data sets from various different technologies. So we tried single cell resolution technologies such as SeqFish, Xanium, as well as subcellular resolution, which is like StereoSeq. And then we also used spot level data sets like Visium. So when we use like the single cell resolution data sets, we characterize each cell as an agent. But then for the spot level data sets, we kind of just said that each spot is a cell for simplicity. However, you could like deconvolve those spots into individual cells using, there's some methods out there, and then you could use our model for that, where each deconvolved cell is then an agent. But for simplicity with like spot level data where it's not at single cell resolution, we just simply made each spot an agent. But it is applicable for technologies at single cell or subcellular resolution as well.
Wonderful talk. I had a question about whether you've gone back and inspected from your results of short and long range diffusion the molecular weight of the soluble ligands under question, because they differ quite a bit. And it would be very interesting if your larger molecules are diffusing less, which would kind of correspond with what you've already inferred for your delta parameters.
So we don't really have the molecular weights. So we just have like the gene. We just use the gene expression data to try to see and like model it, and then just by changing the decay rate and then using the other capabilities of the model, it just kind of like identifies the long and short range. So we didn't really do any analysis on that.
But the corresponding gene product is what's communicating, right? So that's easy to look up. Those proteins, those values are known.
Oh, I see
So
what
you
you mean.
could say, okay, this is a 100 kilodalton protein, and this is 500 kilodaltons. Therefore, their diffusion coefficient should be different.
Yeah.
And then
That,
that would be, because you don't have the ground truth, that would be a really great way to cross check some of your inferences.
yeah, that's an excellent point. That's not something we did, but
Yeah.
definitely something we probably should do in the near future to validate. Yeah, thank you for the question.